So our first speaker of the session is obviously no stranger to MIT, Rick Young, who is a member of the Whitehead, associate member of the Broad, as well as the Koch, and professor of biology at MIT. Rick received his PhD training at Yale and performed postdoctoral research at Stanford. He has served as an advisor for many different organizations, including Science Magazine and the National Institutes of Health and World Health Organization. He's founded a number of companies and been really a presence in that area for some time, and is currently a director of Enzyme Pharmaceuticals. And he has numerous honors, some of which I will mention, from the Chiron Corporation Biotechnology Research Award. He has the Yale's Wilbur Cross Medal, and in 2006, he was named as one of the top 50 leaders in science, technology, and business by Scientific American. But most notably is his recent induction to the National Academy of Sciences. So congratulations to Rick for that. But beyond these numerous accolades, Rick is an all-around good guy and a wonderful and engaging mentor. So I was a postdoc in Rick's lab a number of years ago, and he has certainly impacted my career as well as the careers of many others who have spent time in his lab or who have been a part of his research. So welcome, Rick.
Thank you very much, Lori. I think I'll take you with me to introduce me at other talks. I really appreciate the invitation, Jackie, Tyler, to come and speak with this very talented group. Today, I'd like to focus on the collaboration between transcriptional and epigenetic regulators in control of cell state. And I'd like to focus on two states. One is the embryonic stem cell state, which has given us some of the concepts that have been valuable in looking at the other state, which is cancer. So I'll talk today briefly about how we think of transcriptional and epigenetic control of embryonic stem cell state, and then how that's led us to look at the function of MYC and tumor cells. So embryonic stem cells have been a favorite model system for us because they're great cells in terms of their gene expression program being the foundation for development. That gene expression program is misregulated in many diseases, and so we imagine that as we come to understand how embryonic stem cells are regulated, we may get insights into disease. And it's long been known that there are 4 key regulators that are responsible for driving the transcriptional program via cells. OCT4, SOX2 form a heterodimer, and they're essential really for this key feature, pluripotency. NANOG works with them. It's required to enter and maintain that stable pluripotent state. And c-MYC, in its role as stimulating proliferation and self-renewal, is also a critical transcription factor. These factors have been used in various ways as reprogramming factors by Shinya Yamanaka, Rudolf Janisch, and others. And so they really are not just master transcription factors of pluripotency, but they in fact can reprogram most any cell type into an induced pluripotent stem cell. And in studying how these transcription factors work over the years, we and others have come to the conclusion that the role of these master regulators, OCT4, SOX2, and NANOG, is to basically recruit the transcription initiation apparatus to most of the genes that are going to be active in ES cells. Remarkably, these proteins are expressed at very high levels. Adequate numbers of OCT4, SOX2, and NANOG are available to bind enhancers for the majority of actively transcribed genes. But initiation isn't the whole picture. Once polymerase initiates, it actually tends to transcribe only about 30 or 35 base pairs. It pauses. There are 2 factors that are instrumental in this pause called NELF and DSIF, and they require phosphorylation by a cyclin-dependent kinase, CDK9 cyclin T, also called PTEF-B for positive transcription elongation factor B, in order to license polymerase for effective elongation. And what's become clear from studies in embryonic stem cells done by Pete Rawl, Charles Lin, and others in my lab is that licensing is a consequence of c-Myc mediator and BRD4 recruiting this cyclin-dependent kinase to phosphorylate DSIF, NELF, and the polymerase itself, and thus licensing it for this efficient elongation. So I want you to think just a little bit about how transcription factors and chromatin regulators operate across this embryonic stem cell genome, and I'm going to summarize it in the following fashion. So here's a pie chart of the sets of genes that we call active, initiated, poised, or silent. And at active genes, you have a stereotypical type of behavior. OCT4, SOX2, and NOG are bound to an enhancer. They bind coactivators like mediator. There are switch-SNF types of nucleosome remodeling factors that are brought into the promoter region. Polymerase initiates transcription. MYC, BRD4, brings in CDK9 cyclin T, as I mentioned, to facilitate elongation. And you have a series of histone-modifying enzymes that make modifications that are stereotypical down the transcription unit. So you can actually see the product of the trithorax group, HCK4 trimethylation near the promoter, the DOT1L modifiers a little further down, and SET2 type proteins in the sort of 3' half of the gene. There's a set of genes that are initiated, and they go through all of this process except to stimulate elongation. So these are genes that Christian referred to as non-productive. They initiate but not elongate. And typically at these genes, MYC and BRD4 are absent in the vicinity of the control regions for these genes. There's a set of poised genes, and these are the bivalent genes. We call them poised because they are transcriptionally silent, but they're rapidly induced on differentiation. There's a suspicion that many of these genes see some low level of transcription, and that the non-coding RNAs produced by initiation are involved in some cases in recruiting polycomb. There are other chromatin regulators like SETDB1 that have been implicated in repression of this set of genes. And here again, we can see OCT4, SOX2, and NOG associated with many of these genes, but MYC is generally absent. And finally, this other set of genes, the silent set, is distinguished by having these histone H3K9 trimethyl marks and the absence of all the other modifications and apparatus that's associated with these other class of genes. So as you might expect, the master transcription factors in MYC are absent from this class of genes. So to set up the problem that I'd really like to talk about, which is MYC and ES cells, what we know, MYC in cancer cells, what we know in ES cells is that MYC's functioning predominantly to enhance transcription elongation at most active genes. And we've become quite curious about what happens in tumor cells with deregulated MYC. And the curiosities come in part from the fact that we know that there are about 30,000 molecules of MYC in these ES cells that are driving increased elongation across all the active genes, but we really don't know what happens with what's called oncogenic MYC, or at least we didn't until this group, Charles Lin, Jacob Loven, Pete Rawl, and Tony Lee, together with Ron Parnell and Jay Bradner's lab, and Charles Lin's a shared grad student with Chris Burge, all got together to ask what is going on with oncogenic MYC. So you'll know this, MYC is the most frequently amplified oncogene. It's deregulated in at least 40% of cancer cells, and it's widely thought to be a driver in most cancer cells. Its overexpression generally correlates with poor clinical outcome, tumor aggressiveness, and a variety of other phenotypes that are of interest to us. This is Steve Burley's crystal structure of MYC with its heterodimer partner, MAX. And what struck me when we began to consider this problem is the sheer volume of papers that had previously described something about MYC function. We could find more than 20,000 papers describing some aspects of MYC function in cancer cells. And so this is from Vita and Hendrickson, a review that simply described all of the things that MYC does in a normal capacity and the kinds of things that MYC also does when it's deregulated. And by deregulated, generally we're talking about overexpression. So this is just an incredible spectrum of functions. And basically, almost anything that's been described in biology, it seems that MYC can impact. And it's surprising that it has such a dramatic impact because if you look at MYC target genes, if you look at the signatures that others have described, it's really, there are two distinguishing features. One is that you can identify using expression analysis on microarrays hundreds of genes that are altered by high-level expression of MYC. But as you look from one cancer type to another, you see really a minimal overlap. That minimal overlap nicely includes many genes that are known to be involved in proliferation. And so MYC's widely regarded as a regulator of cell proliferation and as having target genes that are involved in proliferation. But it's remarkable to me then that these signatures really don't explain in any way I understand the very diverse phenotypes that are described for dysregulated MYC in biology. So we decided to look at this problem using cells from Qidang's lab. This is a P493-6 B-cell lymphoma that's been engineered so that you can turn off MYC or you can turn on MYC, and you can control over a period of time then the level of MYC accumulation in these cells. And we also looked at a variety of clinically relevant patient-derived tumor cells. I'm going to focus most of my remarks, though, on this more controlled experimental system. So this slide shows two results, really. So the first result is from ChIP-seq data. When we take these P493 cells and look at the set of genes that are occupied by RNA polymerase II, which is a good measure in our mind of the transcriptional behavior of the genome. What's quite striking to us as we look at where MYC occupies the genome, it's really just occupying the set of genes that are actively transcribed, just like in embryonic stem cells. And remarkably, as the levels of MYC increase in these cells, you don't see a change in the set of genes that are occupied. It's the same set of genes that are occupied, you just get higher levels of signal from each of those genes. And these numbers over here reflect the number of c-MYC molecules that have accumulated in these cells at the times that are being assayed. And so you may remember I said ES cells have about 30,000 molecules. So what you're seeing here is a range from about 13,000 to over 300,000. So MYC is, across this 28-fold or so difference in concentration, is continuing to occupy the same set of genes. And if you look at the set of genes that RNA polymerase II occupies across these three time points, it's largely the same set of genes that it's occupying. So the set of actively transcribed genes isn't changing as you, not in a significant way, as you increase these MYC levels by nearly 30-fold. MYC's in the nucleus, so it's not that MYC can't get in. So here's the 0, 1, and 24-hour time points. These cells have been reported to get larger, and they do as they accumulate MYC. And here's the MYC signal inside these cells, so you can see it's increasing, and it's increasing in the nucleus. So MYC's in the nucleus. And what's happening? Well, the increased levels of MYC are leading to increased occupancy at the core promoter sites where at active genes. So here's an active gene. Here's the ChIP-seq signal at 0, 1, and 24 hours, where you're getting 13,000, nearly 100,000, and then 360,000 molecules in the cell. And if you look across the genome, what you see is the substantial increase in the level of MYC at these core promoter sites at active genes. The other thing you see is that you begin to see for the first time at very high levels of MYC what we call enhancer invasion. The MYC molecules are starting to accumulate on the enhancers. So this particular gene, NNT, has an enhancer in the midst of the gene, and you can see as the MYC levels get high, you start to see enhancer binding, and this is a quantitation across the genome. So across the genome, at active enhancers, what you see is an increased occupancy of MYC at the enhancers. Now, this is the kind of thing you might expect just from pure biochemical principles, as modeled in the following diagram. So at low MYC, Cmyc and max, which is expressed at very high levels in most cells, is binding to conventional EBOX sequences. It's a hexameric sequence that's well known. And as you increase the levels of MYC, more MYC-max molecules begin to bind, and they bind at sequences nearby in the open chromatin. This hexamer is present at millions of times in the genome, so there are sequences adjacent the favored sequence where MYC-max can bind. And then as you get to very high levels, in part because the enhancers loop around in proximity to active core promoters, you're going to get MYC-max beginning to bind the open chromatin that exists at these enhancer sites. So that's the model, and can we get any evidence that's in support of that? And the answer is yes. If we just go back to this NNT gene and we map out the EBOX-like sequences, the sequences that are in bright red here are the canonical sequence that it prefers to bind, and these other sequences colored in pink or more white have hexameric similarity to this ideal sequence, but they are not the ideal sequence. And so if we take these secondary sequences that we would predict have weak affinity, we can do gel shift experiments that are competition experiments where we can take the EBOX DNA fragment, add recombinant MYC-max, this is human full-length recombinant C-MYC-max, and show that in competition experiments, the relative ability of perfect EBOX versus variant EBOX sequences to compete off the MYC-max heterodimer. And those results nicely parallel what we see in vivo at these sites. So what's MYC doing as it's binding more to enhancers and core promoters? Well, it's recruiting more PTEF-B. So this is what we'd expect based on the ES cell work. As you increase the amount of MYC-max at these active promoters, what you're getting is an increased ratio of Pol2 engaged in elongation versus initiation. We can measure this because the initiating form of Pol2 is phosphorylated on the heptapeptide repeat of the large subunit at serine 5, and the elongating form of Pol2 is phosphorylated in that heptapeptide repeat at serine 2. So it's stimulating the recruitment of more PTEF-B, and we're getting more transcripts per cell. So over this period of time, what we can see now using nanostring, where we're getting absolute measures for various RNA molecules, what we see across the spectrum of in this case 75 active and 75 silent genes, is that we get in this case we got a 1.7-fold increase. We've expanded this analysis to several thousand genes, and the increase is more like on the order of 3-fold across all the active genes, and no significant effect across silent genes. So you're getting what we think of as transcriptional amplification. Now, is this just happening in this model system? We looked at a variety of different tumors, multiple myeloma cells, small cell lung carcinoma cells, glioblastomas, and measured the levels of MYC in these cells. They ranged from about 110 to 800,000 molecules of MYC protein per cell. Again, it's binding only to active genes. We can't detect binding at genes that are not transcriptionally active, and that occupancy looks quite high, and the enhancers of these genes get invaded by MYC binding. And finally, with respect to these patient-derived tumor cells, when we isolate different small cell lung carcinomas that have different levels of MYC, what we can see is there's a correlation between high levels of MYC and increased transcription elongation and increased transcript accumulation, just as the model system predicted. So what we think is that MYC is not a conventional specifier. It's not a DNA binding transcription factor that identifies specific genes across the genome for expression. Rather, it goes to genes that have been selected for transcription in those cells, and it enhances transcriptional elongation on top of the level of elongation you get with mediator and BRD4 mediated recruitment of PTEF-B. Now, if this model's true, if all active genes are c-MYC target genes, then what's the origin of MYC signatures? And moreover, if what's happening in these tumor cells with high levels of MYC is simply an increase in the 1.7-fold, 3-fold in the number of RNA molecules of all the active genes, how does that contribute to tumorigenesis? And are there other implications? Well, I must say, I take responsibility for some of this because in early microarray analysis, we and others decided that in order to compare two samples, you have to do some kind of normalization. And typically what you do is median normalization. You take the median signal in one microarray experiment and you compare it to the median signal in the other microarray. And so if you have a situation like you have, as I've just described, where you have cells that have low MYC, and across here we're looking at genes ranked by expression levels. So these are RNA levels, absolute expression. In this model where you have high MYC cells and you're getting higher levels of transcript, 1.7-fold, 3-fold more transcript, this is the starting point. As you do in microarray expression analysis, where you're looking at a fold change in expression and you've done median normalization, here's the fold change that you measure for these genes. And because it's median normalized, you'll find a signature. There will be upregulated genes and downregulated genes. But what I would argue is that this is an artifact of the need to median normalize in a system where you're looking at relative levels and not absolute levels. And so most MYC signatures, maybe all MYC signatures, derive from this strategy. And so I think they're useful for diagnostic purposes. MYC does have an effect that you can read in this signature. For example, the signature is in part due to the relative stability of various messages, but it's not a direct consequence of MYC function at those specific target genes. So what does it contribute to tumorigenesis? And I'd have to admit, at this point, we don't know. You can imagine there may be some advantages to a cell that simply has more of everything. And some of those advantages may be captured in the knowledge that we know translational capacity is limiting for tumorigenesis, and we know aerobic glycolysis is limiting. So just having more of these machineries in a larger cell may facilitate tumorigenesis. What are the other implications? I hate to say it, but I think perhaps 20,000 papers have pursued an erroneous notion that mix a specificity factor and has a specific set of target genes. Instead, we think that it's binding to and amplifying expression of all active genes in that gene expression program in the cell in which it's overexpressed. So that means to us that this may account for this perplexing array of cellular functions that have been claimed for c-Myc. Drugging downstream targets of c-Myc in this model is unlikely to be an effective strategy. And instead, we now think that therapies focused on RNA polymerase II pause control and elongation may in fact be valuable for clinical treatment of many different tumors. And you're going to hear, in fact, about attempts to drug other molecular entities that are involved in POL2 pause control and elongation. This level of control in gene expression is emerging as an exceptionally interesting level of control. For most of the half-century of research into how animal cells are regulated at the transcriptional level, we believe that transcription initiation was the limiting step, was really where all the key regulatory steps occurred. And I'm arguing that that's where you select the set of genes that you may want to express. You can do that. But actually, some of the most interesting biology is at the level of pause, release, and elongation. And so a few examples up here in development. PGC1 is a factor that in Drosophila and an analogous factor in C. elegans actually inhibits PTEF-B in primordial germ cells in order to shut down all transcriptional elongation. So you can get initiation of genes in these primordial germ cells, but when transcription is shut down, the mechanism deployed is the lockdown of the elongation step. In hematopoiesis, Lenzon and others have shown there are specific transcription factors that are critical for developmental decisions that are controlling elongation, not initiation. NF-kappa B looks like it may behave like c-Myc. At least in certain contexts, what it's doing is components of the NF-kappa B family are going to target sites, and they're recruiting PTEF-B to amplify key genes involved in inflammation. Antibody diversity and many other interesting processes look like they're now controlled at this level of the recruitment of this critical kinase. And in cancer, just to finish up, what we often see is altered expression, generally overexpression of MYC, BRD4 in some cases, components of what's called the superelongation complex, which is a complex that contains within it PTEF-B and a cyclin-dependent kinase that's a component of mediator. And so all these molecular entities play roles in the control of this elongation release step. And finally, just to Charles and Jacob, Pete, Tony, and our collaborators in Jay Bradner's and Chris Burgess' lab, thank you very much.